I really want to stress through this talk that we should think about diffractive optics much more than we do. We know that there is a problem with it, which is chromatic dispersion, but you can solve that problem. And once you solve that problem, you have complete freedom. Because when you are in Fourier space using a phase grading, completely transmittive, you can multiplex beams, but you can also control their wavefront any way you want almost. So the sky is the limit and your fantasy is the limit to what you can do in Fourier space. I was lucky enough to now have my own PhD student the last 6 years, which means that I could realize some things that I hadn't really been able to do before, including this camera array, which I'll talk a little bit more about. It was really fun. So again, yes, it's more than 2 times as hard. It's at least 3 times as hard to do 3D, right? So here's an example of, it's a Drosophila developing little embryo. And these are just what's going to be the heart cell speeding, just to show that, you know, this is 3-dimensional imaging. And all of this 3D data is captured simultaneously because we are multiplexing the beam into a 3D image. That was kind of the key there. So, you know, Suliana's lab and a few more labs in the world that are getting more and more visible are really just doing tool development for microscopy in academia, not at institutes and companies, but academic research. And why would we do that, right? So there are many, many reasons for doing that. One is that we want to push the field of optics forward. One of it is we want to deliver imaging to actual use. And I'm in the field of biology right now, so that's what we're doing. We're trying to find tailored imaging systems for biologists. And we want to disseminate this. So once you've made this invention, I would say the hardest step is, you know, how do you provide that to the community? And there I'm still kind of wondering what's the best way to do that. I just included a nostalgic photo from the lab when I was a student with Mats and Reto Fiolka and Hesper, and back in the days when structural illumination was something the lab was working on, and I was just building my first 3D imaging system. So what we can get in microscopy, we can get so many things. We can get 3D volumes, we can get high resolution, we can get super resolution, we can get high speed, we can have living cells and nearly physiological conditions. We can have all of these different things, but we can probably not have all of those at the same time. So what I've focused on is really high-speed 3D imaging. So to do that, we need strong signal. There's nothing to it. If you are going to image a whole 3D volume, you are going to need a lot of photons. You're probably going to need custom optics. You're going to need excellent data management and data analysis, because if you take a stack of images, that's more data than if you take one 2D plane. And this is really just expensive and difficult, but it's also really beautiful. And people spend a lot of time alone in dark rooms putting components together. These are some of the students I've worked with through the years with their different imaging systems. We've done a lot of fab work too, because diffractive optics, you can design them, but then you also have to make them. And in a sense, it's easier, right, than refractive optics, because if you want to make, for example, microscope objectives, they're made in France, sorry, they're made in Germany and in Japan. So even France, which is a great optics nation, you don't have that. Even the US, which is a big science nation, doesn't have that. But diffractive optics, you can learn to make hands-on. It may take a couple of years. It took a couple of years for me, but it's really, really fun. So in the US, we have these open nanofabrication facilities that are really for semiconductor physics, where you can also go and process glass. And in France, you have a bunch of these too. I haven't visited one of them yet. There is something on campus that I don't even know what they have, but I know that the Planète in Marseille, you can access and do these things. So diffractive optics are really beautiful things. I brought some diffractive optics here. We're going to talk about what you can do with them in a little bit, but just so you see what we're talking about, it's pieces of glass. So when you do diffractive optics, you can do amplitude gratings. Here's an amplitude grating. It's kind of easy to make. It's a chrome layer on glass. Or you can do phase gratings that are transparent. I can see you through this grating. And that's the real beauty of it. So I'll send these around so that you can like touch them and look at them. And these are not very exciting. They just multiplex a beam if you look at them. We used to have some really fun ones, but I think my students have all of them, little Pokemons. So here I send the beam through this grating and I get all of these orders out. Now I wanted to make this grid, but I could also have made an image of a dinosaur or whatever I wanted, right? So this is just to illustrate that I'm getting all of the light through here, and I can choose the distribution. So here's the amplitude rating with Cruz and the phase. Don't cut yourself on these because they might have sharp edges. So that's what diffractive optics looks like. And oh, why? My slides are in the wrong order, guys. I'm at my microscopes already here. I got ahead of myself. This is just an example of one of the imaging systems that we've commissioned at UC Santa Cruz. And here's more 3D data. So this is truly 3D, simultaneous 3D imaging at full resolution of the optical microscope. These are two freely moving C. elegans animals. They have panoronal expression of GCamp. The color is just the Z projection because it's really hard to do these 3D visualizations. When we get better at that, our talks will be much better. So this is coded in depth with Z. So 3D imaging, I would argue it is really, really cool. 2D imaging is fantastic. So we have the F64 group, right? So their solution to the 3D imaging problem in photography was to get a really deep depth of field, which means that I can image everyone in this room from the back of the room to the front of the room, or everything up in the high Sierra from the mountains in the background to the little mountain lake in the foreground. But even though this is 3D information in a sense, everything is focused at the same time. So I can't really tell what's where. So if we do the opposite, we open our aperture. We have a very short depth of focus. Now we can start to discern between the depth in the image. So here is Cherry Blossom in Central Park. That is a movie that's supposed to play. It's not playing. This one is at least playing. So now I'm stepping in Z through the sample to get that 3D image. So we didn't hurt any flower in this experiment. It was on the tree. So you can see that it actually moves a little bit. And that's very realistic when it comes to microscopy too, because your sample is moving. And if you're trying to scan by refocusing through the sample with a very, very thin focus depth, you may, even with really fast galvos and scanning system, run into the problem that your sample is moving, which is why a lot of imaging is done in 2D. So how do we capture all of that at the same time? We need a trick. So Harold Edgerton's trick back in the days was flash strobe photography that was done even on film with a strobing light. So all kinds of innovations are needed. And when you do 2D imaging on a 3D moving sample, you can use statistics to extrapolate what that little guy is doing. But we really wanted to do that differently. So this was my PhD project that Mats gave me as a task to solve. And that is, let's collect the whole focus stack, all of those Z-planes in one shot, but without sacrificing the optical resolution. And it turns out that you really can do that. Some people try to do that using light field techniques, where you go a step further and say you actually want to capture the whole light field. This is gorgeous. You put a Schack-Hartmann sensor either in the Fourier plane or in the image plane, but it does sacrifice resolution. So we didn't want to do that one. It's great for photography. It can be great for depth ranging. It hasn't been used that much in microscopy, but you can use it in microscopy. What I did instead was to pick up on something that Alan Greenaway had once said, which is that if you put a off-center Fresnel lens in a microscope, you sort of get this effect. You get a multiplexing of the beams and you get a focus shift. So I went to Edinburgh and visited him, and he said, yeah, but you know, it doesn't really work that great. And you have the problem of chromatic dispersion. So we never really did anything with it, but you know, great if you want to try to make that happen. So I did. And diffractive Fourier optics is really about the wave nature of light. This is sort of what I was explaining to the students in the class this morning, that light really is a wave, and diffraction is something we don't work with. We work reflection. Okay, I'm reflecting something in the mirror. We work with refraction. We have lenses, but diffraction is much, much harder. And it's making its way around that little diffractive grating there. It's really neat. So what's on that little piece of glass is one of these little shapes. And this little grating here, this little diffractive optic, it's simultaneously multiplexing the image of the microscope and refocusing it. So how does that work? Well, we have a microscope. What is a microscope? It is an objective and a tube lens. The objective has a short focal length, the tube lens has a long focal length, and therefore we form a magnified image. Now, if I put the diffractive grating in my laser pointer, I get a spot in, I get multiple spots out. If I put a diffractive grating in the Fourier plane of my microscope, I get multiple images out. Now, these images are really truly identical. They are dimmer because I'm splitting the light between the different images, but I'm filling frequency space. I am not compromising my resolution. So if I take this image, I still have diffraction limited resolution. The whole pupil, the whole Fourier OTF support is maintained. So that was step one here, right? We want to multiplex this image. Now we want to fill it. The camera chips are usually square. So let's create a grading function that gives us an array of images that like this. So here's a 3x3. That was all we dared to try to fit on the old camera sensors. The grading function here, it looks like that little goblet or little fan. People call it different things. It's made by the algorithm that I wrote. It's probably the worst code you've ever seen in your life. It's written in MATLAB by a grad student who doesn't like programming. So it took actually 5 days to run on my old plastic Mac laptop. It's much faster now that my student has rewritten it in vectorized form without for loops in MATLAB. I recommend doing that, but it really works quite nicely. So what you do is you want to find the grading function that gives you the image you want. The image you want could be a guitar. My Japanese student made a Pokemon. I wanted 9 spots. So I take a random guess, then I flip 1 pixel in that guess, I Fourier transform those two, and I compare them to my target function of 9 spots. And the one that's better, I keep. And then I keep stepping through and flip the pixels and flip the pixels and flip the pixels. And it looks something like this. So I'm flipping the pixel and I'm flipping the pixels, and all of a sudden I've got my 9 orders with even intensity. I'm even suppressing the 0 a little bit for a reason that we'll talk about later. So that's a phase grading. So if you want to design a diffractive grading for fun or for science, you can download the pixel flipper and you can give it the target function you want. And if you want an amplitude grading, that's easier to make, where you just make that chrome mask and you transmit and block light. You put MATLAB, you give it a 0 for block and a 1 for transmit. And if you want that phase grading, a binary phase grading, you give it -1 and 1 for 0 and pi phase shift. So that's the pixel flipper algorithm. It's kind of beautiful. The pattern is kind of beautiful too. That gives you 67% photon count intensity effectiveness. If you want more, you're going to have to do some more complex things. You're going to have to do multi-layer grading. You get an even more beautiful shape if you allow this optic to have multiple stair steps in the glass. So that's 3 etch layers of 8 steps. And then you get 89% photon count efficiency. I think it's amazing. It's really, really, really beautiful to make those optics. So that was the multiplexing. So now we can, with 89% efficiency, multiplex light into 9 images. And we want to focus shift them. So how do we focus shift them? We apply a distortion to this grading. If the grading is periodic, all of these beams are going to come out with the same angle. If we introduce a distortion in the grading, in places where the grading is coarser, the light is going to have less of an obstacle when it's squeezing through that aperture, right? So it's going to diffract a little bit less. If you have a tight spacing, it's going to diffract more. So I can control this diffraction angle. And why do I do that? What happens to my beam? What happens to my beam is that this beam that came in flat is now diverging. That's the +1 order. The -1 beam is converging. And this, if I calculate it properly, I can make that into a perfectly aberration-free refocusing function by calculating the out of focus phase error for each of those focal planes according to the Aberration condition, and then just programming that into that grading function. And now I get that focus step. So the math for doing that is to calculate that out of focus phase error, and you get an equation that you can separate in X and Y and apply the distortion to that grading. So now we have it. We have simultaneous 3D imaging. We have a focus stack of planes without losing resolution that just can run like that onto a single camera. The problem with this, and the reason that people don't use diffractive optic, is chromatic dispersion. The person who's sitting and squinting and looking through that grading right now is probably going to see a little rainbow. That rainbow is so strong that even green GFP light, maybe 30 nanometer bandwidth, is going to be a green rainbow. So we need to fix that. And this is the reason that people don't use diffractive optics. They're beautiful, they're light. Like that little piece of optic is doing so many things, but the dispersion is killer. And you're going to get this smear out in the image. So the first approach is, okay, let's put in a second grading that reverses the effect of the first. So if you make a blazed grading that has like little prisms as its pattern, that is going to send 95% of your photons into the -1 diffractive order. So let's do that. Now we have an issue with this because what happens is you completely reverse the effect of the first grading, so they all end up in the center again. So in order to fix that problem that we just introduced, we add another optic, which is this prism. So this is the incredibly beautiful, difficult to make, complicated, lovely, simultaneous, full resolution 3D imaging system. And it's great. We have now managed to eliminate that prism, and I'll talk about that. That is not published yet, because if you let your PhD student graduate and take a job before they've published their paper, they're not going to have time to publish their paper after that. So later tonight, I'm going to talk on the phone to my PhD student, who is now at the Chan Zuckerberg Biohub with the notable colleagues of some of you, Loic Royer and that whole team. But we will eventually publish it, but I'll talk about it tonight, today anyway. So that was really hard, and it took a long time, and applying it to biological problem was one of the many things to do. So what you get is those 9 planes, and then you can track whatever it is you want to track with it. That's yeast centromeres. This is transcription factors. And then what happened is that I ended up at Rockefeller University. So then, we've been looking at these tiny things like the cell nucleus, 9 focal planes. Actually, the grading you have there, it even has only 7 spots, because one of my collaborators said, Sarah, this is a waste of photons. My first and my last plane, they have nothing in them. Can you make just 7 planes? So we made just 7 planes. But when I got to Corey's lab, the problem was another one, and that was that they're looking at animals. They're looking at, okay, they're small animals, but they're much bigger than the cell nucleus. So the idea here was that we would try to make one of these microscopes with much more focal planes. So first we tried it out, and we saw, yeah, it looks beautiful. These are the sensory input neurons from the worm. It's smelling almonds, so some of them are turning on and some are turning off at that. But the idea was that, if I run my pixel flipper again, and now I want 25 spots, that works too, and I get this grading function. I don't know if you're into jazz. There's a jazz conservatory here. This makes me really look at, like, that's the Frank Zappa logo of his beard. I don't know if anyone here is old enough to know who that was, but that's the grading function for this microscope. So we did this silly proof of concept. I talked about chromatic dispersion. The reason I could image this was I put a laser line filter in my emission channel. So this is 1 nanometer bandwidth of fluorescence. That did not work to image like that, but we just wanted a proof of concept. Another issue was that this is 1 single camera sensor. Fitting 25 planes on a single camera sensor was maybe not the best idea for your field of view. So I figured, okay, one day I will have my own lab. And I really didn't want to have my own lab. I wanted to be a postdoc forever. It was amazing. It was really great. I knew that if I became faculty, I would have to do all these things. And even if we love teaching, if you have to teach full time, I mean, I knew how bad it was going to be. So I stretched my postdoc out as long as I could. I wasn't in HH my lab. It was great. So I said, yeah, okay, I'm going to have to start my own lab because I want this to be done. And I'm going to need to have my own lab for that. So that was how that happened. So there is one thing about this though that it's not playing. If you don't have chromatic dispersion correction, you're imaging monochromatic light. And when you think about bright field, you think about white light. But bright field doesn't have to be white light. You can put a laser line filter in your white lamp, and you can do bright field with monochromatic light. So we did some embryonic imaging at the Woods Hole and also in Corey's lab with this 25 plane single camera microscope. And I think people should do more bright field imaging. I saw some bright field imaging experiments in here today in Alexandra's lab, right? And yeah, it's gorgeous. So that is really simple to build. So if anybody wants to do that, all you need is that little piece of glass that you're passing around, and you just put that into your Fourier plane, and then you can do bright field imaging. If you want to do fluorescence, it's a lot more work, but it's a little less work now because of this revolutionary new design for diffractive optics chromatic dispersion correction in a camera array. So you see in front of every camera here, they all have little lens mounts. So you put a little lens in the camera, and the lens has a little filter mount. I don't know how many photographers there are here who shoot with regular cameras. So in that little filter holder, you put one of these blazed gratings that I talked about, where each little grating feature is this little prism-like thing. And then that's a rotatable mount, so you just rotate it in the right direction. And now you can skip that prism I showed. So now you have a chromatic dispersion corrected diffractive optics imaging system with just that pairs of grating. So I think this is going to be super useful. I already talked to a colleague who is doing a compressed imaging application without this focus shifting grating, with just multiplexing the beam and overlaying planes. And you can use exactly this camera array for that. So this camera array, this is consumer technology, right? This is just machine vision cameras that you mount next to each other. And then you have that optics that you made in the fab together with your students, or maybe your students now can do them on their own. So this was, we call it the M25. It's really after one of the messier objects, because we also love astrophotography. But that's how that looks. And again, this prism now is eliminated. And that was actually the hardest part to make. The diffractive optics are easy to make compared to the prism. I couldn't even make it myself. I had to buy it from someone who custom made it. So you see here how each of those image beams hit that grating, is corrected for the chromatic dispersion, and then focused by the camera lens itself. And this could be done with 3x5, with 5x5, maybe with 6x6 cameras, who knows? So here's Eduardo, who built the microscope. He's a wonderful guy. He is now at the BioHub in San Francisco, and we're trying to get him to publish this. I think it would be really nice if he would do that. He did some really beautiful imaging. This is beads. So this is aberration-free refocusing over 50 microns. So if you try to do refocusing of something without moving the objective relative to your sample, you're going to have massive spherical aberration, because the objective is only designed to be aberration-free in the focal plane. And I think this has to do with the fact that the Herschel and the Abbe condition cannot be simultaneously fulfilled unless you do some kind of remote focusing, or this type of aberration-free refocusing in Fourier space. So 50 microns of aberration-free refocusing. And the first time we did this, we actually didn't get it right, because we had to change the distortion, because it has to do with the refractive index matching too. And then, yeah, they imaged some beads dancing around in water, and that looked great. They got 25 cameras running at 160 hertz. And after 2 years of soldering and the PSOC controller boards, they can actually do that without dropping frames. So we did some proof of concept imaging at the Woods Hole Marine Biological Laboratory, which is a wonderful, wonderful place on Cape Cod. These are just still images, but they're dye-injected neurons in lamprey and zebrafish, because that's what they happen to have in the lab. The very first movie, here the frames are not synchronized yet, was with Muscle G-Camp in C. elegans with Jennifer Morgan and Gail Haspel. Even after seeing this, they had patients with us, and they kept imaging with us. And we also imaged these paneuronal C. elegans. So the hope is now that, in order to publish this, we could just publish this as a method, but we would really like to track the activity in all of those neurons we're probably going to give up on that. I asked a few people who are really good at image processing and said, yes, Sarah, that is actually really hard. Maybe you shouldn't do that. And you should just publish this method. And then you can just put the data somewhere, which is a nice tradition in my lab that we always publish our raw data. And someone else can download that raw data and just track it for you. So if someone wants to do that, please go ahead, because it's really hard for us. There's also data on these muscle activity. So here Ed has tried to divide the muscle up into segments and look at what happens. And you can start to see these opposing signals in the worm body as it's wiggling and moving freely in water, not in space. So that's what, that's another panoronal movie. We also imaged some Drosophila because our friend Thomas had Drosophila. And here is one of those times where we really want to stress that, sure, if you do light, if you do like Scape or some kind of light sheet thing, that can be really fast too, right? It can be probably almost as fast as this, but it also has the amazing advantage of selective illumination. This is wide field illumination. You're illuminating everything at the same time and you're imaging everything at the same time. So all the out of focus photons are lost. But you can do bright field with this. And I think that's powerful. And I think we should do a lot of correlated imaging with bright field and fluorescence. You can use the bright field if you're doing, for example, developmental imaging of little embryos or something that are super sensitive. And if you want to put some fluorescence in there, you can put that too, and you can image that also when you need to. So that was some of the Drosophila heartbeats. So the raw data is on the left there, the 25 planes. And then we've tried to make a 3D projection on that. We're getting better at it. We're not super good at that yet. And the user interface for this, the students wrote in Apari. So far, I have never come to the lab with a sample and they have been able to start the instrument. So we're still working on the user-friendly list. There's also always oh, but it was just this little thing. Yeah, sure, there's always some little thing. But maybe one day someone will be able to do that really, really well. Encasing the system is crucial, right? We want to have a little body. We want to have a irregular microscope body, of course, with the eyepiece is super important for biologists to find their samples. Some days finding the sample is the hardest thing you do all day, right? But at some point, in order for this, it's a long time ago since I invented this technique. I tried to build some systems for people who wanted them. It has to be commercialized. It's just something that needs to happen. It needs to be made robust. I'm gonna try and make an experiment and work in industry at a company that makes binoculars and see and learn what is it that you actually need to do in order to design a system that's robust, that you can screw in, because if you want to have one of these on your microscope, you just take the camera off and you put this black box in and you put the camera at the back of the box. That should be it. But for that to happen, there's a lot of very careful engineering that needs to happen before. So my hope is that I will now try to learn that. And there are people in the imaging community who, they start their own companies, and I'm not an entrepreneur, I'm a scientist. It's fantastic that they're able to do that. Sandrine Levesque started a company 7 years ago. She's got 40 employees, they're selling instruments. I'm never going to do that, but I'm hoping that I can at least get someone else to commercialize this. It would be really cool. I wanted to make a little note, since I'm from Mats's lab, it wouldn't be complete without a little bit of sim. That movie is not playing. This is one of my favorite movies in microscopy. It's Lin Shao's movie of mitochondria with 3D sim. And you can see the cristae, and it's 20 seconds per frame. That was not fast. Now things are much, oh, come on. Sim, again, improves both resolution and contrast, and it's resolution in 3D, not just 2D. So it's a really beautiful method. It's spatial frequency mixing. You have a spatial frequency of your illumination that mixes with the frequency of your sample. So all of a sudden, these little guys through moire patterns arising can be seen. And you do a little bit of reconstruction, and you move these components out in Fourier space in order to reconstruct the super resolved data or higher resolved data. And this used to be, I used to have a slide for that, but I guess it fell out. This used to be a challenge because this software was not very easily available. And then Marcel Miller in Bielefeld decided to do his PhD on writing an ImageJ plugin for sim reconstruction. So now if you have sim data, you can just drag it into ImageJ and go through Fair Sim, and it can reconstruct the data for you. I did it today for someone on my laptop. That is a really important step forward for people to be able to trust sim data, because you can get artifacts when you do image reconstruction. But if everyone who publishes really cool data publishes their raw data, and if you can go in and reanalyze that data yourself, you're going to see if it's real or not, right? So I think that's super important. So a single frame of 3D sim takes 15 frames. If you do 2D sim, it's only 9. That can be done really fast now. Oh, this only plays once. So 25 hertz with the spatial light modulator with super fast pattern translation and rotation. But if you want to do that, and here's an example of reconstructed data. This is my favorite data set ever. It's the synaptonemal complex of the mouse spermatocyte. So it's the two sister chromatin strands that are wrapping around each other, and they're about 100 nanometers apart. So you really cannot resolve this with wide field. But with sim, you can just resolve it, and you can see how they turn around each other. So that's like one of my favorite examples. So if you want to do 3D imaging in sim, like for example of that synaptonemal complex, if you want to label some proteins between those two sister chromatids and look at the dynamics, for 9 focal planes, you need 135 time frames. That's a lot. So of course we put sim and multifocus together and made the multifocus sim microscope so that you get, at least you get the 3D information in one go. And this is what that system looks like. It's very simple. It's just the multifocus on the emission side and the sim on the illumination side. And we built one of those in the imaging facility in Santa Cruz. And there's a little bit of proof of concept, synaptonemal complex imaging going on. The reconstruction becomes hard here, and Lin Shao is working with one of my students on trying to solve that. And then hopefully, if we can do that in a useful way, we'll be able to image this live. This is so far fixed samples. So that's one of the future goals of that. This system has also been built in Stockholm, but by one of my collaborators. We brought his student Max over to Santa Cruz, and then he duplicated the system in Stockholm. So he has been looking, of course, at actin, since that's really popular. So I'm hoping that he will carry that forward and keep working on that too. That was all I had to talk about today. So thank you again for having me here. It's been really cool to see new people and old people and other people who came here from California. Yeah,